Thank you very much. I hope you'll forgive the pun, particularly at the expense of the competition. So the second half of that sentence is of the title is the most important bit, of course. And I just want to give some context. I'm a clinical consultant. I'm not a bioinformatician. I'm not a nanopore whiz. I'm somebody with a clinical problem. And it's important to recognize what, as a neuropathologist, what pathologists we're doing. We're trying to predict, on the basis of a tissue sample usually, what the patient's future holds. We're trying to put them into categories, because that's the basis on which they're treated. And as you'll know, we've used microscopes for a very long time, but increasingly we're finding that we know that the molecular changes are key. And there are lots of reasons for that, and time constraints mean I can't go through this in any detail. The hours I'd love to talk about this for, you're saved from. But you can see at the top right there's a patient's brain scan that's got a tumour in it. And a few years ago I diagnosed that tumour, and at the time I knew I was getting it wrong, because I had a sense that this was an entity, but I didn't know what that entity was. Thanks to the array, we now know that that's a particular tumour that's defined by a fusion. But there's no way I could have done that with a microscope. Now, the current model of testing for molecular testing is not ideal. It's based on when the system that we currently have delivers the answer. It's not when the patient needs their answer, it's backwards. And so this is the reality for me in Nottingham. We had to send cases to London, ringed for those of you who aren't so sure on where London is. And as you can see, there's a timeline here that ends in 28 days, and it's often longer. And if you're a patient or a patient's family or a clinician waiting for an answer, waiting for a diagnosis, then there are obvious massive implications for not having that data when you need it. So driven by this, we've developed an ultra-fast pipeline that is based on this P2 solo thing and some novel steps that we've developed. DNA extraction, library prep, and then very rapidly, based on that methylation fingerprint you heard about so beautifully this morning, we can classify tumours in an intra-operative timescale, because the first step of a patient's treatment isn't when they meet the oncologist, it's the surgery. And if we can inform surgery, then outcomes will be as good as they can be. So within 2 hours, we have an intraoperative classification, and then over the next few hours, up to 24 hours, we get the rest of the data. So fusions, SNVs, single gene methylation, copy number changes, etc. Now, we really are standing on the shoulders of giants, some of whom are in the room today, to do this. So we're using multiple classifiers. And again, the caveat, I'm not a bioinformatician. Professor Matt Luce will take your questions later on this. But in short, it's incredibly powerful to use multiple classifiers that use different methods to generate an answer. And this is a case that a colleague reported the intraoperative smear on. Unfortunately, he was having a bad day and got it wrong, but within 5 minutes of sequencing, we knew exactly what this tumour was, that it was metastatic melanoma. And we've done this on a large number of cases now, and again, you're saved from me going through the detail of this, but we get a very, very high level of concordance. And those cases with a standard of care, those cases for which we don't get concordance, we understand why. We haven't had a single case for which we've got an answer that we don't understand, a wrong answer that we don't understand. Now, intraoperative is great because it's really funky, it catches the attention, and we initially thought that we would probably only use this in a relatively small number of cases, particularly complex pediatric tumours. But talking to neurosurgeons, and I'm afraid that's something I have to do each day, now they know that this is available. The increasing repertoire of tumours that they think this will help is enormous, and it's actually which tumours wouldn't you want an intraoperative classification? You know, if you're making intraoperative decisions, neurosurgery, shocker, it's not straightforward. It's not just an operation, there are nuances to it. So if we could give them really comprehensive, confident data for which to base their intraoperative decisions, then that would be ideal. So I'm just going to take you through a few cases just to demonstrate this, a couple of cases. So this is an 11-month-old child who presented very recently to us. I don't need to point out that tumour to you. I did the intraoperative smear, and it was a very primitive-looking tumour. So there's a very wide differential of tumours that this could be. I gave that diagnosis, and the plan from the surgeons was to have a subtotal resection. So those deeper bits of tumour that are in the slightly more hairy areas, they decided to leave and await the standard of care diagnosis. Now, several weeks later, we got the confident molecular diagnosis that this was an ependymoma, a tumour that really needs to be resected fully. So that patient then went on to have a second operation. And that's really unfortunate, because we knew within 5 minutes of sequencing that this was an ependymoma. But not just that, we could tell you exactly which type of ependymoma it was. And this is one of the bits of software that Matt's developed for us to visualize these things. You know, pathologists, surgeons, we're all visual people. Understanding what's going on is really, really helpful. If we'd been able to tell this diagnosis, which we got several weeks later through standard care pathways, intraoperatively, that child and their family would have had a very different outcome. And of course, there are system efficiencies to be had from using this sort of technology. As I was saying, we give results when the system delivers them, not when the patient needs them. And this case really highlights that. So this is a 3-year-old child with another tumour I don't need to highlight to you. A very short history, so a short history makes you think it's going to be an aggressive tumour, and this one certainly was. I did the smear on this and had a differential for what it was going to be, but couldn't give a confident diagnosis. That's just the nature. On this occasion, that's not a deficiency on my part. That's just the nature of the work that we do. But we knew, again, very quickly that this was a particular type of tumour. It was a group 3 medulloblastoma, and that's incredibly useful to have intraoperatively. But the other thing with this pipeline is that we're not just delivering intraoperative data, we're delivering all the rest of the data over the coming hours. We get copy number, as I mentioned, and this demonstrates that there's a gain at chromosome 8, and if you zoom in on that, that's in the locus of the MYC gene. Now, MYC is an independent, MYC amplification like this is an independent risk factor for this particular type of tumour. So this is the conundrum that we have. Through our research-only pipeline, within 24 hours, we had a confident diagnosis as well as risk stratification. Several weeks later, we get the standard of care data, and I'm still actually waiting for some of that. We had all of the data that was needed to treat and counsel the patient within 24 hours, but we can't share it yet. And so increasingly, we've got this ethical dilemma of we're actually potentially doing harm by not sharing this data, and that's something we're trying to wrestle with at the moment. Now, neurosurgery, some grim pictures, I'm afraid. That's the neurosurgeon, not the operation site. On the left, this is an open resection, and the neurosurgeon here has applied the melon scoop and delivered us a great big bit of tissue, which is fantastic. Now, when we started doing this work, I was very concerned about the amount of tissue that we'd receive. We're now doing it on stereotactic biopsies, and we can get confident classification on roughly 1 millimetre squared of tumour. Now, of course, there are caveats to that about tumour cellularity, etc., but we've had exceptionally good results on really tiny bits of tissue. So we are very much building on the work of others here, but this Robin, which is the visualization software I've shown you bits of, has a number of strengths to it. Really using multiple classifiers. The idea that one classifier is which is the right classifier, in our experience, is probably the wrong way of looking at things. Using the multiple classifiers is really powerful, because in some cases one classifier gets the answer first, other cases it's another one, but knowing that each of those 2, 3 or more classifiers is giving you the same answer gives me, as a pathologist who's trying to join all of these dots up to make a diagnosis, increased confidence. Having the visualization of the dynamics of how the diagnoses are appearing tells me things. It also gives the neurosurgeons things to bet on. But it's incredibly helpful when you've got complex samples that contain a mixture of control and tumour tissue. Now, we're fortunate to use the P2 Solo, and the amount of data and so on that we get from that gives us added confidence. We're aiming for speed, efficiency, confidence, and this pipeline gives it to us. The novel ultra-fast library prep I haven't talked about in any detail, but that's key to it as well. And then using the adaptive sampling, we've got inbuilt panel sequencing. We have a gene panel of about 250, 300 genes that we're including in this. But on occasion, when we've come up with a rare entity, we know that there's a particular fusion or gene that's involved in it. We can then add that in on the fly and identify that fusion, which then gives us more confidence. You've got the methylation classification as well as the fusion, for example. So I'm out of time. This is the work of many, many people, as you can see here. We're not the most diverse group, but there are, and apologies for the beard, it's before I realised that, like my late mother, a beard doesn't suit me. But if you would like to find out more, here is a QR code. We're running a day on the 27th of June in Nottingham, and you're all welcome to come and find out how it's done, because we don't want to be doing this for the world. We want to be having these little bits of kit in neuro-oncology centres all over the place with a pipeline that delivers the equity of access for genetic testing that our patients need.